Our next presenter will be part of the student competition. Mrs. Shaimaa Khandaker will present a job on investigating the exposure to analgesics and the impact on the metabolism during pregnancy. You're welcome.
Thank you. Good afternoon, everyone. Thank you all for having me here. I'm super excited to be a part of this event. So my name is Shaimaa, and my PhD research is looking into investigating the exposure to pharmaceutical drugs and their impact on the endogenous metabolism during pregnancy. So my supervisors for my PhD research is Dr. Luke Wiley, Dr. Nicola Gray, and Dr. Sam Lodge from the Australian National Phenome Centre at Murdoch University. So I'll start off with speaking a little bit about the prevalence, the use of drugs by pregnant women. So about 60% of pregnant women take at least one or more than one medications, and about 70% of lactating women also take some form of medication. However, the bigger problem here is that 90% of these medications lack labeling information. Now, the use of medication during pregnancy has actually increased over the past decade, and the total prevalence of medication usage is even higher when considering over-the-counter medication. Now, this over-the-counter medications also pose the risk of self-medication, and currently the worldwide prevalence of self-medication amongst pregnant women is about 44.5%, which is relatively high. Now, the current pregnancy categorization system also raises further concern. So FDA and TGA actually considers paracetamol to be quite safe by ranking them at A and B, which means it's a low-risk drug. Now, the way they come to this conclusion is by doing animal tests, but the results they get from the animal test is only with acute dosing. And it is understandable that pregnant women are a vulnerable cohort, which is why they lack clinical trials. However, there is evidence that there may be adverse effects. So really, the current knowledge gap today is how the chronic exposure to common medications such as paracetamol influences maternal metabolism and offspring outcomes. So evidence-based research findings also raise further concerns. So if we were to, I've just given an example of two papers amongst many. So if we were to look at the figure on the right, so this was a paper where they've done human observational studies on a pregnant woman population, and they've also followed up with the child outcome. And they've actually found that there is an association of prenatal paracetamol exposure, also known as acetaminophen, APAP, with reproductive and neurobehavioral abnormalities such as ADHD and autism. Now, the paper on the right is a key paper for my PhD research. Now, this was a very large study where they've actually applied both metabolomic and metagenomic analysis on a non-pregnant population. And they've actually found that with the use of paracetamol, there was a significant depletion of sulfated sex hormones, which are also known as neurosteroids. Now, neurosteroids are vital for healthy brain development during early fetal stages. So really, the study also further questioned whether the wide use of paracetamol during pregnancy may actually play a role in hormonal homeostasis. Now, my overall research aims and method design for my PhD consists of three aims. So my first aim will be applying data mining to pre-existing study cohort NMR data. Now, for the first aim, I'll be using the nuclear magnetic resonance spectroscopy, which is a metabolomics platform that we use to analyze our samples. And with the aim one, I'm interested to data mine on different classes of drugs, which are very commonly used during pregnancy, including analgesics, antibiotics, and antidepressants. Now, in parallel to this, I'm also developing a targeted method, which is my aim two. And the targeted method that I'm developing is on endocrine signaling molecules such as steroids, lipids, hormones, microbial metabolites. And then further on, in aim 3, I'd like to validate and apply this targeted cohort on the birth study. So, and for that, for aim 2 and 3, I'll be using liquid chromatography mass spectrometry, which is also known as LCMSMS, which is the other metabolomics platform that we also use to analyze our sample. So today's talk will be focused on the first aim. So I've only completed my PhD. I've just completed my first year earlier in May. So I'm just going to talk about the first aim, where I have some preliminary findings. So with that, as I said before, I have applied data mining to look for paracetamol in a pre-existing NMR study cohort data. Now, NMR actually has quite a few advantages. The fact that it's non-destructive analysis is very quantitative and has high resolution structural information. It's very reproducible and requires minimal sample preparation. And you only need one experiment for both targeted and untargeted analysis. Now, some of the research questions I'm interested to ask in this aim is what the prevalence is, what the reported versus measured is, which usually is quite different, and also what the demographical differences are. So for today's talk, and my current investigation has been applied on a study cohort from Sydney, Australia, known as MUMS, which stands for Microbiome Understanding Maternity Study. Now, this was a longitudinal prospective observational study where I've analyzed both serum and cord blood of about 100 pregnant women participants, and I've looked at 4 different time points, including first, second, third trimesters, and birth. Now, just before showing my findings, I thought I'd just share how we actually look for the paracetamol metabolites. So this is, if we were to look at the 2 figures on top, the first one is actually showing NMR spectra of healthy urine, which is blue. And the second one is showing NMR spectra of healthy urine, but after paracetamol intake. Now, the third one, which is at the bottom, is when we overlay both before and after paracetamol intake. So which is showing both blue and red. And we can clearly see that the peaks there is the paracetamol glucuronide, which is one of its conjugate, and that's the paracetamol sulfate. Now, from here, we can also see that paracetamol glucuronide can be found in higher concentrations than paracetamol sulfate, and that's common in blood and urine. And so we did further investigation with paracetamol glucuronide. Now, one of the very crucial steps before we actually find confidence that, yes, we are actually looking at paracetamol and not something else, is we need to make sure we remove any overlapping metabolites, which are found in the same region as paracetamol. So, for example, I've used serum, so obviously there would be quite a bit high content of protein. So in this case, phenylalanine was overlapping with APAPG, and so we had to make sure we removed that. And if you notice the noise spectra in the background, which is in purple, so we always try and put a threshold on that, because that's just noise level. So anything above that is when we are confident that, yes, we are finding paracetamol. So with that, this was my initial findings. We found APAPG in serum and cord blood of this MUMS cohort. I've looked at trimester 1, 2, 3, and birth. So orange is representing serum, and blue is representing serum cord blood. And the dashed line is the noise level. So anything above the dashed line is when we're confident, yes, we are actually picking up paracetamol conjugates in these participants. So clearly we can see from trimester 1 to 3, there hasn't been many. But then most of paracetamol use has been found in birth. So about 23% was found in serum. And even 4 participants, we actually found paracetamol conjugate APAPG in cord blood. Now, one of the things we also like to do is individual differences, just to see if there's any sort of pattern or any sort of like correlation. But clearly, it was very different. Each participant was very different from one another, from trimester 1, 2, 3, and birth. However, I was curious to look closely into the participants where we found APAPG in cord blood. And there was about 4. So the first 2 participants, we found APAPG is present in serum and cord blood during birth. The third participant, we found APAPG is found in serum during trimester 1, and both in serum and cord blood during birth. And the fourth one's quite interesting, because we found paracetamol APAPG in cord blood, but not in serum. And so hopefully we'll do further investigation into that. So to summarize my talk today, we can clearly see that paracetamol exposure is very common during pregnancy. However, with my investigations, I understood that it is more common during birth than the other trimester 1, 2, and 3. And the fact that APAPG paracetamol was found in cord blood, it clearly shows that the fetus is exposed to drug metabolites. And as I mentioned before, past studies have shown association of prenatal exposure to paracetamol with neurodevelopmental disorders such as ADHD and autism. And also there is further concern is also raised with the past study, which showed the frequency of hematopoietic stem cells was found in cord blood was diminished after paracetamol use. So with that, I'd like to end my presentation, and I'd like to acknowledge all my supervisors and my colleagues over at Australian National Phenome Centre for their ongoing support. I'd also like to thank MUM's study cohort for giving me the opportunity to analyze their valuable cohort. And I'd also like to thank Brookers for funding my scholarship. Thank you.